protocol observed. Good evening, ladies and gentlemen. This talk will indicate why I love microbiology, what we have achieved together with my students, and where we are headed. So enjoy. Why microbiology? This is the reason why we're here today. Specifically, why bacteriology? Often I get asked this question, and the answer for me has been the same, even today still the same. Bacteria are interesting to me because of their simple structure, simple genetics, and their dual nature. While some are beneficial, some are, however, pathogens. This complexity is what makes them interesting to study. And microorganisms or microbes, they form one of the biggest life on our planet. They are widespread, found in almost all environmental niches, and bacteria form the largest group of these, not only in terms of numbers, but also in terms of diversity. To date, we have named and identified above 43,000 bacterial species, but we've only validly following the rules, published just above 24,000. And if you have a look at this picture here, bacteria are the largest, but we've only validly published and named just above 20,000. And with this, it's argued that our knowledge on bacterial species is quite minimal, which is currently standing at below 5%. And this is because we only study a few selected species, which could be because of their importance in health, in the environment, or their applications in various industries. And what I have here are the top 20 most studied bacterial species in 2024. And it's no surprise that E. coli is the top, because this is the model species for all bacterial species. We know about bacterial species because we have studied E. coli quite significantly. And species like Vibrio cholerae, which is the one that causes cholera, will only receive attention at the times when we have outbreaks. And as you can see, in 2024, we only published, well, not me alone, but everybody around the globe, around just above 10,000. So for us to have a comprehensive understanding of bacterial species, research efforts must be focused beyond just a selected few species. But how does one go about studying bacteria? Everything that we do, we follow rules, we follow principles. And within bacterial species, or the study of bacteria, simply put, taxonomy of bacteria. Perhaps before I dive further into this, I should explain what taxonomy is. Taxonomy is the science of classifying and naming species. In this case, bacterial species. And we do this based on their characteristics, which could be how they look, their structures, their morphology, their biochemical structures, and even their genomic material can assist us in checking if the species are closely related to each other or not. And that is how we classify and name them. In 2018, a proposition was made that those who are interested in studying bacteria must include genomic material in there, which meant we go beyond to study the genomic material and look at a few parameters that could assist with determining if a species is a new species or it's related to other species. Chan and colleagues were correct in making the proposition because we are within what is called the second era of molecular biology, wherein we're not only able to sequence genomes within a short space of time. And I think I might have said a word that I might have lost some of us in here. Genomics is the study of the entire DNA material of a species. And with being in the second golden era of molecular biology, we able to sequence the genomes these days within a matter of hours. And we can do it today, tomorrow we've got the results. And this is simply because of the advancements that have been done in the sequencing technology. So with all this positivities, with all these good things that I just mentioned, where does one begin when they want to study bacterial species? As I speak, I am taking out quite a significant number of them. And whoever you're sitting next to now, they have their own associated with them. Some are found associated with animals, some with us human beings, and some with plants. And within my research group, we specifically look at bacterial endophytes, which are bacterial species associated with plants. And these endophytes provide significant support to the plants, assisting it with nutrient uptake, as well as production of what are called phytohormones, which are simply hormones that are needed by the plant for growth and development. And in return, the plant provides an environment for the endophytes to survive. When I joined UJ back in 2016, I established the Bacterial Genomics and Biotechnology Research Group, and we sought to study bacterial endophytes specifically from medicinal plants. And this is because research had indicated that the endophytes played a role in the production of the metabolites by the plant. So we sought to understand the traditional uses of the plants, which called for us to start with indigenous communities in the country. This is where our research always starts. We go there to understand what the uses of the plants are, because the indigenous people here are the ones that use the plants. And in addition to that, we also try to identify and get to know the types of bacterial endophytes within these medicinal plants, as well as the role that they play in plant growth and development. And beyond that, through genomics, that is genomics of the bacterial species, we then look at all the different genes, all the different types of genes that these endophytes code for, and possible functions. And the specific ones that we look at are what are called biosynthetic gene clusters. And these are simply a group of genes that are co-localized together, and they produce secondary metabolites, which are simply compounds. And these compounds have bioactivities such as antimicrobial and so on. So what we do beyond that is to check if this metabolites from the endophytes would be similar to that of the plant. You know the saying, if you've got the gene, you've got the protein. Further to that, we check what the possible applications of these endophyte species could be. How then does indigenous knowledge systems tie into the work that we do? As I indicated earlier, that our work always starts with indigenous communities because of the knowledge that they have, the wisdom that they have with regards to these medicinal plants. And bridging the gap between indigenous knowledge and modern science, it offers guided discovery, ensuring that we have enhanced scientific relevance. At the same time, as scientists, it forces us to have, or rather decolonize our approaches to research. And this is the best way to foster an ethical and inclusive research. And this, by the way, is just a tip of the iceberg when it comes to indigenous knowledge systems or incorporating indigenous knowledge systems into modern science. So what are some of the discoveries or achievements that we've had within my research group, the Bacterial Genomics and Biotechnology Research Group? And I say our discoveries because these are not my discoveries alone. They are my discoveries together with my students and the great University of Johannesburg. So, in this article here, we looked at bacterial endophytes, focusing on those that occur at the aerial part of the plant, Pelaicalomelanus. And together with SPIWE, we utilized morphology as well as data backed up by sequencing of the 16S ribosomal RNA gene. And we were able to identify, I would say, a few species. And some of the key discoveries that we made here was that over time, some pathogens, that is over evolutionary time, some pathogens would turn into bacterial endophytes. But then the question we asked is, what then would be the differences between a plant pathogen and a bacterial endophyte, which I will answer later on. In addition to that, we also looked at some of the compounds or secondary metabolites produced by this bacterial endophytes, most of which showed antimicrobial activity. And this aligned with the plant's use or application. By the end of 2021, we had done several similar species, except that we were not recovering a high number of bacterial endophytes. Then we sought to look at the entire community through metagenomics. And with Dr. Maela here, she recently graduated, we looked at the whole bacterial community endophytes within Dicoma anomala throughout the 4 seasons in the country. And what we discovered here was that soil type, season, as well as the host plant, specifically the host plant, will dictate or have an influence with regards to the number of bacterial species or the number of bacterial communities. So we discovered that late autumn, including winter, there is quite a significant number of bacterial species, as indicated in the circle on the left, and on the right is just an indication in terms of the genera species. So this was actually proof that metagenomics does result in recovery of more species. And then we pressed on. By end of, well, not by end of, as of last week, we have officially did about 28 whole genome sequences, but we've only published 25. And this is from around just about 6 medicinal plants. And we have identified, either using the whole genome sequence or single genes, about 80 species. And we still have more that we are still busy with in the lab. So there's 2 circles here. These are genomic maps or genomic profiles of two of the endophytes that we have worked with. I can't reveal the names because we're still busy with the work. This is as far as I will go here. And at this point, I would like to stop and acknowledge my student, Mr. Spamandla Msomi, who has assisted with generating this Circles preferred genomic maps. And as indicated here, what they show are the different kinds of genes that they code for. And beyond that, we can get to see the diversity as well as ascertain the taxonomy of the species. But of importance is the fact that these genomes can also assist us in finding out the functional capabilities of these bacterial endophytes. So what discoveries have we made with regards to the genomes? First off, earlier on I spoke about a pathogen and an endophyte. If we say that an endophyte is an endophyte, it must be able to support plant growth. What then would be the difference between an endophyte and a pathogen? The one significant difference are what are called the virulent genes or the virulence genes. And these are simply the genes that are required in order to cause harm. Pathogens have more of those, whereas endophytes have less of those. And other things like plant cell wall modification, these both would have. And this is simply because they both need to gain entry into the plant. In addition to that, I spoke about earlier the phytohormones, which are simply hormones needed by the plant for growth. We have identified in endophytes significant numbers of genes that are responsible for coding for phytohormones such as auxin, salicylic acid, ethylene, and many others, which are required for plant growth and development. So any other takes from the whole genome sequencing? Further to ascertaining the biotechnological applications of the species, we can also are able to check if a species is a novel species or not, which makes it even more exciting. I have students who are sitting here today. Some have isolated useful enzymes from these endophytes. Some have isolated and currently working with novel species, and some are undertaking research from spores, which I will also speak about later. So there is quite significant benefits from genomics-based research that can offer solutions in a variety of or different industries. What we have also discovered is the fact that incorporation of other omics research, such as metabolomics, which is Prof. Madala's speciality over there, incorporating metabolomics, transcriptomics, together with genomics, we do have comprehensive and meaningful outcomes. So I've indicated that we have, at the moment, officially 25 whole genomes of bacterial endophytes. And these vary from genus to genus. In around 2018, we sequenced a bacterial species called Pantoi, and we identified a gene therein called the TCCZ gene, which is simply a gene that codes for a protein with insecticidal activity. So this protein is able to kill insect pests. So what we then did to try and mass produce this gene together with Prof. Kondia, who's seated over there, we tried to clone this gene within E. coli. The cloning aspects, we succeeded. The E. coli got transformed, but we could not get it to express the protein or express the gene rather. And I think by the 8th or the 9th month, Musibudi, Miss Malomani here was like, no, I think we've done all that we could. And I am mentioning this here today because this talk for me is also a platform to reach out to a possible collaborator who can come on board, and we see on how to improve on this. And before I speak about this, I have a wonderful group of students, and I have the pleasure of showing you their phases as I go through this. Here we have Dr. Diale. She's also from Limpopo. And what she did here was to look at the whole genome of the species, but at the same time, we went to the lab and said, okay, these are the genes that it codes for. Are we able to get the strain to express those genes in the lab? And that is exactly what she did successfully. In terms of the genome, she identified quite a significant number of what are called bacteriocins, which are simply peptides with antimicrobial activity. They are able to kill, yes, kill will come quite often, but if we are killing the pathogens, we are winning. So they're able to kill pathogens. And in addition to that, she also identified quite a high number of enzymes, which are simply carbohydrate active enzymes. So the carbohydrate active enzymes can be used as a marker to determine the lifestyle of a particular species. In this case, she identified a high number of genes that play a role with regards to colonizing the gut. And by the gut, I mean that of animals as well as us, possibly human beings. And identified also a high number of genes that play a role in biofilm formation, as well as degradation of oligosaccharides. And all these pointed us to a probiotic. And since we had the data genomically, expressed it in the lab, that is in vitro, and the young generation would say they gelled together, supported each other. They were congruent with each other. And I'm happy that we have since partnered with Prof. Bilani, who's sitting at the back over there. Prof. Bilani is an anatomist as well as a physiologist. And we've also are partnering with Prof. Kennedy at West University, my alma mater. And in this particular partnership, we are now looking at, will this potential probiotic have any effect in zebrafish? And if it does, then we will move on. I can't reveal where we move on to, but Executive Dean, more money will be needed. When we started this work, I never anticipated that we would land on novel species. And we did. And when we did, it made it fascinating, it made it interesting and worthwhile. Cindy here, who is about to submit her PhD thesis, isolated a novel species from Dichoma anomala, and through morphological data, biochemical, and specifically genomics, we were then able to ascertain that this is a novel species, which meant at the time, I think it was between 2018 and 2019, that's when we had the strain here. And we just found out, she came running from the 7th floor from the lab to my office on the 2nd floor and said, please look at my results. Does this make sense? We had a look at it, and I said, yes, it does. And my next question was, do you know what this means? And she said, yes, we've got a novel species. And we marched on, brought in more collaborators, Prof. Mutadi and Dr. Chweni, I believe they're in the audience or online, and also brought in Dr. Liu from China in trying to fully characterize the strain. And as you know, with discovery research, when you discover something novel, you get afforded the pleasure of naming it. And we decided to go with the name Bacillus dicomyi, which at the time that we published it was the 20th member and the first species within Bacillus series group to be described from the African continent. And that has definitely ticked a box for us, and we still have many, many more boxes to tick. When I joined UJ in 2016, Prof. Ngulunga, I used Bacillus series, which is a species isolated elsewhere in the world, to teach micro ones about bacterial spores, which are simply spores produced by bacteria. And today, Dr. Sobola, who is sitting over there, she is using Bacillus dicomyi. Well, I can't teach micro ones because I'm now the head of department. She is using Bacillus dicomyi, and it makes it even more fascinating for our students to learn that Bacillus dicomyi was discovered by scientists here at UJ, and even more better, by a student within the great University of Johannesburg. So this is the best way to decolonize microbiology, and it has indicated that South African plants and many other species out there, they can and they do play a role in microbial global diversity. She accidentally, those of you who know about how penicillin was discovered, it was by accident. Here, another accident happened. She was growing the species for production of spores and accidentally left it over the weekend. And on the Monday when she came back, she made observations and she was like, but this looks like crystals. Crystals are other proteins that are insecticidal or kill insect pests. And at the moment, only Bacillus thuringiensis is the only species globally that produces these crystal proteins. I would say this is a possible newest discovery as well, but I will get as far as I can with this because we need to get publishing and create more knowledge. Further to that, we've looked at the lipids, the fatty acids of the strain, all of which play a significant role in other various industries. And beyond that, we've also seen from the genome that the species does have a significant role in terms of plant health, support, and biotechnological innovation. This picture here on the left indicates to you the genomic map of Bacillus dicomyi. And the circle there, well, it's not a circle, it looks like an oval, yes. That oval there indicates the region that we are currently working with. Yes, we have fully here at UJ sequenced this genome, although it appears to be fragmented. So that means we need to go and polish it up there so it can look quite solid like the rest of the circle. And I am happy that we are now have partnered with Dr. Ben Pascoe at University of Oxford, and this new partnership will see Mr. Spelele sitting over there going to spend about 4 months at Oxford, and positive and wonderful things will come from here. And on the right, it might look like rice, right? It's not rice. It's the actual Bacillus dicomyi that we observed under the phase contrast microscope. And those little lights looking like structures, that would be the spores. So you've got the cell and then the spores within there. And Prof. Ngulunga and Prof. Mutete, if there is a group of scientists on this planet that must know and understand Bacillus dicomyi to the core, it is myself and my students at this wonderful university. And we will embark on that. And more money is needed. And I'm happy to also mention further that in trying to understand Bacillus dicomyi in its entirety, we've also partnered now with Dr. Ntimango. Yes, he is over there at my alma mater at Wits University. And with him, we are looking at the different metabolites that this species produces. And we will take it further and so on from there. Is our research impactful? Does it have any societal impact? I think for the fact that we have discovered and described a novel species, and it is the first one within the Bacillus group that speaks into the quality of education that we offer. And earlier on when I started, I indicated that microbes are found in almost all environmental niches within our planet. And that means for us to achieve all the SDGs, research, whether you like it or not, will have to include and focus microbes because they affect us in every possible way. And as I conclude, I know often when someone speaks about microbiology, people either think of germs, but today we know that they're not just germs. It is either a pathogen or it is a good bacterial species. And with regards to microbiology, when you speak about microbiology, people will want to, they just think about microscopes, but we have went beyond that with my research group, combining classical microbiology with genomics. And our research and many others have indicated that bacterial endophytes indicate or are untapped reservoir for possible applications in a variety of industries. And we have seen, including many other studies out there, that integrating indigenous knowledge systems with cutting-edge research, in this case, genomic tools, results in quite significant discoveries. At the same time, it offers us an opportunity to validate and respect the knowledge from our indigenous communities. This results in decolonized and inclusive, sustainable research. And together with my students, we march on, and we are remaining committed to collaborative, ethical, and transformative science. And perhaps I should stop at ethical. Prof. Mutete, as a botanist, will know that in this country, South Africa is part and parcel of what is called the Nagoya Protocol, which is there to try and protect the biodiversity of the country, in terms of the species themselves, but also in terms of the digital sequences that as scientists we utilize. So that means if we are to use a plant, before I even go and speak to the communities, I need to get permission. Where do I get permission? From the government. And we get permits for that. To describe a novel species, one needs to have what is called an export permit, because currently we do not have an internationally accredited or recognized culture collections lab. So we need to get an export permit for that. That is ethical, and that is how we do our work. So what work is awaiting us, or what are we moving forward to? We have isolated enzymes, we have isolated antimicrobial compounds, and the next step for us is to commercialize these products. Ching ching, Prof. Mutit. More money. And we will do this looking into other aspects of research. We've already done it. We're already doing it with Prof. Madala as well, including metabolomics and other omics research. And we will strive to improve on our genetic engineering, Prof. Kondia. And at the same time, still going back to where the research starts, which is the indigenous communities. So far we have worked out, say, with officially 6 plants, unofficially 9. So South Africa has how many medicinal plants, Prof. Motete? It's quite a lot. So we still have a long way to go. And as we do this, we remain committed to policy ethics, ensuring that the work that we do offers access and benefits, not only to us, but the indigenous communities where we start, which is the reason why I am also a member of the Digital Sequence Information. This work would not be possible without my wonderful and excellent students, and they are all here. Some of them are joining online, and I just want to put a disclaimer. If your face is not here, I didn't get it on time. Yes, because we are meeting every Tuesday, and that will be the first question I get asked. If you are not here, you didn't send it on time. I have seen the commitment of these students, and I continue to see it, and there is still more for us to achieve. And this group of young people have left a quite significant mark in my life. If there was ever a time that I saw their commitment, it was during the pandemic. We couldn't come to the lab, but we made efforts to meet online. And some of the discoveries that I shared with you here today, it's because we continued with the work. And when there was no longer lockdown, we just hit the ground running. And let me first start by giving special thanks to my God, my Lord and Savior, in whom all things exist and consist. It is by His grace that I'm standing here before you today. And I would also like to thank my mom and my dad. I've always watched other people in other platforms receive honors and awards and say, I would like to thank my mom and my dad. Today is my day. Allow me to honor my mom and my dad. You might see a professor standing here, a full professor that is standing here, but these are the two people who have seen all of it. They have made significant sacrifices for me to be where I am today. Mama and Papa, thank you so much for instilling in us the importance of education. I speak not only for myself, but for my siblings as well. And I hope that me and my siblings, we will teach the same values to our children. So thank you so much. And in extension, I also want to thank my grandmother in absentia. She's the one I grew up with, and education for me started with her. By the time we were living with our parents, she had already laid the foundation. And I want to thank my entity number one, my husband. Thank you so much for your support at all times. He is another person who sees the breakdowns, who sees the lows, the highs. And when I'm low, he will say, "Come here, what do we do now? Where to from here?" Thank you so much for your support, my love. I really appreciate it. And and to our children who are watching Cocomelon at home now, well, I had hoped they would watch me on YouTube, but yeah, we'll see when we get there. They always jump when we say, "We just published a paper." And I believe that this video will one day make clarity to that. And my in-laws, thank you so much for your support, for checking on my babies when I go on research conferences, on research meetings abroad. I really appreciate that. And to my besta bombas, the 1,000 team, they know themselves. These are my siblings. And my cousins, countless of them. Prof. Ngulunga, if I was bringing the whole village, you would have to get a much bigger venue. So I want to thank each and every one of you for being here. Boragadi, Bomalomi, Bomrata Malomi, Boramogolo, Bomangwani, Bomamogolo, Kamoka, Galena, Kalabor. These are the people who have raised me, and I have grown up with them. And for those of you who might not be aware, both my parents are teachers, and they have taught me. My mom played a significant role in my formative years. She taught me how to write and read. And by the time my father came in high school teaching me biology and English, Mama had already laid the foundation. And I am grateful to the both of them. I also want to give thanks to my other teachers who I believe are watching online. Mr. Mochawa, Mr. Tladi, Mr. Moraba, Ms. Moshugwa, and many others. Thank you so much from the bottom, if the heart has a bottom, from the bottom of my heart. I have seen you make wonders with the limited resources that we had in our school, in order for myself and many other learners to believe in science. And I also want to thank all my friends who are here. Some are joining online. Thank you so much. I really appreciate that. And I've already thanked my students, right? Yes, I must thank them again. Thank you to my students one more time. I want to thank Prof. Vincent Miles Gray, who was my PhD supervisor, and he remained supportive even after I had left his lab. Actually, he forced me out of his lab because I went to him after I had submitted, after we received my PhD results, and I said, "Prof, I think I have a project for postdoc." And he said, "No, not with me. Go somewhere else." And I'm glad that he cut that cord, because had he not, I don't think we would be here. Thank you so much, Prof, for your support at all times. And I also want to thank Dr. Pilelane Mpangase. I believe he is joining online. This is somebody who taught me bioinformatics. To do genomics, you need to have bioinformatics knowledge. And at the time that we thought it was difficult to learn and attain, he made it quite simpler to understand. So thank you so much for that. And there's a saying in Zulu that says, "Indlela ibuzwa kuba phambili," which means if you are to undertake a journey, you need to ask those who have walked it before you. And I am grateful to my two wonderful mentors, Prof. Kenneth Mwenda and Prof. Glenda Gray, who are both joining online. I really appreciate the wisdom and the knowledge that you share with me, and challenging and encouraging me to dream beyond where I am. Thank you so much. I also want to thank my coach, Mr. Moroing, who is here as well. Thank you so much for assisting me in my leadership journey. I really appreciate this. And there is a wonderful group of female academics that I always go to, not only about work, but about personal matters as well. And they have become sisters, friends. Thank you to Dr. Nunu, Prof. Nomugongo, Prof. Mabuba, and Prof. Sondes. Thank you so much. And the A-team at the great University of Johannesburg, the Department of Biotechnology and Food Technology. It is a privilege and an honor to lead this wonderful team. It is not by chance that we are #1 in the whole country with regards to food science and food technology. Thank you to each and every one of you, from the students, from the staff, the postdocs, the undergrads, and postgraduate students. And I also want to thank my former line manager, Prof. Green. He is still supportive to this day, this very moment. Thank you so much, Prof. And the executive dean. It is so wonderful to serve under your leadership. You make it worthwhile. One does not dread DCM. One does not dread coming to your office. Thank you so much, and may God bless you. And I also want to thank the HODs group. Most of them are here, some are joining online. This group, hey, thank you so much for this group to be a support for each other, to learn from each other. And those of us who are young like myself, we learned from the ones who've been at it for a very long time. Thank you so much. And I also want to give thanks to my collaborators, both local and international, from the global south to the north, from the east to the west. We collaborate in order to advance capacity development. We collaborate because it is fun to do so, and it's even best to find like-minded people to do it together. I want to thank each and every one of you for being part of my journey, part of my research. I thank you. And research is impossible without money. It is true. We need money to make the discoveries, and I want to thank all our funders listed here, including the great University of Johannesburg. Thank you so much to each and every funding agency listed here. And I want to thank each and every one of you for being in this auditorium, taking time to come and listen to me. And I want to also thank those who have joined online. May God highly bless you. Thank you.
Thank you very much. Let me start by sending my greetings, VC Factionary, Professor Ngulunga. I'm from Venda, so it will be difficult for me to pronounce this name. And my former colleague, who's now the Executive Dean of the Faculty of Science, Professor Anna Mutete, who was asking me earlier on whether if I'm still wanting to come back at UJ. And I said, of course, because as you have heard, this is a great university. My greetings extend to you all, family, friends, colleagues. As I've already mentioned, I come from a great province where my sister comes from, and most of you, and most of the students who made these discoveries. So I can't say a great university because that name has already been taken by University of Johannesburg, but we will copycat it. We are getting there. So as I've already greeted you, you are greeted in Madekwan. Firstly, let me congratulate my sister and colleague, Hope, for this wonderful presentation. Most of you, this is a great achievement by her and her student, and indeed, it is a testament of how a professor should be. This is what we are looking for when we are decolonizing this education. We want to see people like you leading the line, and we are ready to follow you. So, I'm here to give a response. What is the response? Maybe for some of you who are not scientists, I've heard you screaming, clapping hands, but maybe I should take it the other way and examine you to see whether if you understood what she was talking about. But let me spare you that moment because I don't want to embarrass you. So, however, I am here to simply simplify this for you and also maybe give some advice and encouragement to my sister. So what she was talking about is something that has bearing on you. You'll hear why. Let me start here. This presentation kept us seated there with amazement of all the publications, all the discoveries. But the question is, what does it mean really to us as human beings? So, in response to this wonderful lecture, I'm thrilled to share some key takeaway home messages that will surely shape how we think about microbial world around us. We can all agree that the story of endophyte microorganism is both compelling and astonishing, both from scientific and social point of view. Let me begin by reminding you that we human beings, we rely on microbes. I'm not quite sure if you are aware that plants, or rather animals like ourselves, like cows, they don't eat grasses. You might have seen a cow grazing a grass, but it does so with the help of a bacteria. Without a bacteria that will help it to digest that particular grass, it won't even eat it. It doesn't like it. The only way it's eating it is relying on the bacteria inside it in order to help it digest that particular grass. A good example for you is a gut microbiota. Inside our gut, from here in our mouth all the way to the stomach, there are bacteria, and these bacteria shapes who we are, our well-being, our immunity, the way we digest food, sometimes even our mood. Those people who are called to have mood swings, it's because of some of the bacteria inside your body that affects how you behave on a daily basis. So you can therefore appreciate this bacteria that you are sitting there healthy and happy because the bacteria are happy. So today, there's even what we call microbiota transplant. What do we mean? For instance, if someone is able to digest and process food faster, let's say a skinny person and you want to lose weight, in some developed countries like in Europe, they are able to take the bacteria from the gut of a skinny person and give it to someone who's a little bit chubby, and as a consequence, that person can even lose weight. So, at my home university, I have a colleague who's called Professor Yoshan Moodley. He works on the association between a bacteria that is called Helicobacter pylori, which is found in your stomach. This bacteria has been with us since time immemorial. So this bacteria has been found to have been with all the people since the migration of the first person who left Africa 60,000 years ago. So the bacteria has been co-evolving with us. Unfortunately, as Hope has mentioned, some bacteria are good, some bacteria are bad. This one is a bad bacteria because it's the one that causes ulcers. However, as it has been noted by Professor Moodley, this bacteria has been evolving in a way that whenever our genes are changing, it's also changing with us. And what he has noted is that some people who have moved away from here to Europe, Asia, they possess different forms of this bacteria. In so doing, he's able to trace the lineage at which you have evolved. West Africans, Southern Africans, Central Africans, depending on this bacteria that is in your stomach, one can tell your origin. So you can see it's not just a mere cause of ulcer, it's a bacteria that speaks of your own identity. In the same way, today's lecture has demonstrated that plants too exist in close association with microbial partners, a relationship that holds enormous biotechnological potential. As many scholars have predicted, the next pandemic, she spoke about the pandemic, that we were suffering COVID-19 not so long. The next pandemic, as it has been predicted, it is going to be antimicrobial resistance. What do we mean by antimicrobial resistance? We mean that you'll go to a doctor with a simple cough, and he will then have to tell you that there's nothing they can do, you can go back home to die, because there will be no medication to treat that cold. It is already happening in hospitals with diseases such as tuberculosis, where people are dying from a bacteria that 20 years ago could be treated. Similarly, you could just have a cut and a bacteria will infect your wound and you'll go to a doctor and the doctor will tell you there's nothing we can do. A simple cut can kill you in the future if nothing is done. Therefore, the work that Professor Hope is doing is to, again, preempt what will be happening in the future so that solution can be achieved now before this pandemic hits us. So, as many scholars has predicted, the next pandemic could be driven by antimicrobial resistant bugs, which are pathogens that no longer respond to known therapies, particularly antibiotics. Where then should we look for solution? The answer, as we have seen today, to plants. Our people have relied on plants long time ago, but Prof. Hope is taking it further and say, let's also look at the bacteria that are found in these particular plants. In this association between plants and microbe, is this association between plants and microbe a new science? Yes and no. We have long known that some plants depend on symbiotic microbes. For instance, those that assist in nutrient sequestration. So already we know that the climate is changing, isn't it? There are droughts. If some of you who are here looking beautiful, you are from Limpopo, we are all pretending here to be happy because there's aircon here and everything. But everybody from Limpopo knows very well that our temperatures can hit 45 degrees Celsius, especially for us up north in Venda. So we are going to end up in a situation where we are going to have excessive droughts. These droughts are going to prevent us to grow food. So we need something to circumvent that. I'm not sure whether if you are aware that there are plants that grows in a desert where there are no rain for more than 5 years. How do those plants survive there? It is also found that some of those plants, they exist in association with some endophytic microorganism that Professor Hope was saying. So scientists are going to the desert and harvest those plants and their microbe and give them to our crops so that our crops can survive drought. So you can see the importance of this thing. So, but recent research, including that of Professor Hope, has revealed something even more exciting. Many endophytes can produce unusual, previously unknown chemical, some with great values. I'm here to help you to discover them and characterize them. I want to give you an example. There's a plant in Amazon called Aqualaria plant, which when infected by a certain fungi, this fungi it produces the resins inside the plant to produce something called oud. Some of you might know oud. Some Prof. Mutete, you were sitting next to me. If you had smelled nice perfume, I'm wearing oud today because of that reason. So this is the perfume that is made by a fungi that grows inside a plant. It's one of the most expensive perfume. You find it, yes. So you find it mostly in the Arabic world. So you can imagine, we can see the bacteria that are telling us about our origin. We can see the bacteria that can help us grow our plants. We can generate therapies from them. We can produce perfumes from them. Beyond perfumes, many endophytic microorganisms are now being shown to produce compounds with medicinal potential, including anti-cancer activities. Allow me to share an example from my own research in Venda. Some years ago, I think 3 years ago, one of my students was working on a plant that we eat in Venda called blackjack, Bidens pilosa, Professor Mutete. For some of you who grew up in the upmarket, Johannesburg and all that, Bidens pilosa is not a food, it's a weed. But for some of us in Limpopo, it's food. However, there's something striking about Bidens pilosa in Venda. You know, Venda is very mysterious and magical, which some people say is witchcraft, but this plant, as we eat it as food, others use it as a contraceptive or to induce abortion. But we always wondered, how can a single plant have good and bad activity as a food and also as an inducer of abortion? But lately, we have found, indeed, through indigenous knowledge system, through consulting the elders, that there is a time of harvest for that particular plant. There's a specific season where that plant harbors a fungi in it, and that fungi produces a chemical that mimics estrogen in women. It can confuse your menstrual cycle, and then you cannot get pregnant if you eat that particular plant. This is something that we gathered from indigenous knowledge system, which further reaffirms your position that you have taken, Professor Hope, that the future is through indigenous knowledge system. Consulting the elders, because you really said it very well, that in Zulu, the Zulu saying, in Venda we say, Ndira ibudziswa ripanda. Let me continue. So, building from this lecture, it is important to note that future hold incredible possibilities. When you started, you mentioned that you put a chart there where you put list of bacteria that are used as model bacteria. And I want to tell you that according to me, as a respondent, today you have to listen to me, that the reason why we are faced with some of the long-lasting challenges is because we have been stuck with Escherichia coli. Prof. Green, we must move from Escherichia coli. Dr. Tlo, let's forget about Escherichia coli. We have had enough of Escherichia coli. We have new challenges here. And your work is doing so well in discovering new bacteria so that we can discover new things, because the future that is coming requires new things. From your work, I can already see that new therapeutics are going to be developed. We are not only going to discover new bacteria, but if you continue in this trajectory, I can assure you that one of the diseases that has always been a problem to us through this work we are going to find new therapies. Because these bacteria that she's isolating are not just mere bacteria that she's sequencing. In collaboration with all of us, some of us who knows how to identify chemicals, we are going to identify one that we are going to develop in a form of a pill. Remember, I'm simplifying this for you. You must really thank me. So we are going to develop through her a pill that you can treat a certain disease, which otherwise, if without her, that doctor was going to tell you, go home and die. I continue. The integration, and I want to emphasize this one, and I'm not saying this because I'm from Venda. The integration of indigenous knowledge system is the future of your work. You, of course, when you say a plant is medicinal, you don't just go to the forest with a closed eye and pick one plant and isolate a bacteria from it. There should be prior knowledge of what that particular plant does. And I can already attest to you that that information is there with the elders. Please continue to go to them, find out from them, you will discover great things. I want to close by telling you, Professor Serepa-Dlamini, there are still plenty of microbial treasures hiding out there. Go and find them. Thank you.
Now, to the fun part. Professor Motiti, if you may please join me, and Professor Serepa-Dlamini, we are going to do creation. I don't know how do the yes. Now, ladies and gentlemen, let me congratulate you, Professor Serepa-Dlamini. Now, you are truly a professor now. I mean, I think they have seen you being inaugurated, inducted. Congratulations once again. Now, there is something that I didn't do at the beginning. To congratulate you, your better half, to congratulate him. Now, I don't know, I mean, how do you deal with a spouse who's a professor, I tell you. Man, all I can say is to say, God bless you. But congratulations. You know, in our culture and tradition, when it is a wonderful ceremony like this, we sing. But I do not know, because normally now at our graduation ceremonies, we sing Sianamaren. But you see now, I don't know what you do for professors. I don't know what you do for professors. Professor Motiti and I will go and consult and think a bit. I think we do need to compose a song for professorial inaugurations. And I wish to thank all of you for taking the time to witness this wonderful, wonderful ceremony. I tell you, I do quite a lot of this, but this is one of the best professorial inaugurations that I have done. And thank you very much, Professor. Now, I don't want to keep you for long. There is food on the other side and drinks. And I can tell you, I'm one who's eager to go and indulge a bit. It's been a long day. And on behalf of our Vice Chancellor, Professor Litlokwa Mpedi, I really wish to thank you for coming to this great University of Johannesburg. And I was listening to Professor Serepa-Dlamini. She said she studied at, what do I say now? At a mining school across the road. But you redeemed yourself though by coming to work at you redeemed yourself by coming to work at the great University of Johannesburg. And I congratulate your students for coming to study with you. I wonder how it feels to be a student at the other side. But ladies and gentlemen, please do join us for refreshments. And thank you very much for joining us. Thank you. And congratulations, Professor.